Started. Thank you. That's Ruben. Ruben who keeps remembering to actually record. I always forget. And today we have John Hodge, and needs no introduction since he's a previous host of this seminar. So, John, I'll turn it over to you. Thank you.
Thank you so much. Awesome. So today I'm going to be talking about some work that we just wrapped up, looking at variation in stomatal spatial patterning in maize. And uh, just to kind of provide a brief overview about some of the topics we're going to go over, I'm going to walk through the idea of using stomata as a model system for thinking about complex traits from a developmental perspective. I'm going to introduce this new spatial patterning phenotype that we ended up developing based off of some ML annotated phenomics data. And once we kind of have that information, we're going to explore it through a couple different ways, either through structural equation modeling, as well as through QTL mapping, so that we could try to think about how some of these genetic effects can sort of cascade through trait spaces. So before we get started, though, although I do love maize and maize genetics, I am classically trained as a botanist, and so this is my botany tax slide. And I really do like to, sort of think about these broader questions about how plant lineages can sort of arise across diverse environments and face a lot of pressures. Where if we were to just walk through a number of different ecosystems, we could have a environment that's kind of a blast furnace. We could have another environment where it's incredibly dry. We could be in sort of a mesic Midwestern prairie that had just burned, or we could be in a sidewalk near you. And across all these different habitats, we're going to see a variety of different plants that are occupying these niches. However, although we might not sort of think that there's a parallel to maize in some of these ecosystems, in some cases, they're actually uncannily similar, because what we're actually seeing in one of these, if we dig through the leaf litter, is that this is actually Tripsicum dactyloides. And so we're seeing a very close cousin of maize that's sort of recovering from this fire, either through regenerating from its rhizomes or depositing seeds into the seed bank. Uh, so there's a couple different ways that plants can sort of respond. One of the ones that a lot of members of my lab like to think about are these behavioral or physiological responses. And Joe Crawford in my group just published this lovely paper where he was able to actually watch stomata opening and closing as a example of this sort of like active movement process. Uh, there is also the capacity where passive movement is also possible. So if you ever think about leaf rolling or bulliform cells, that's actually what you're kind of seeing taking place, is you're seeing this passive capacity of the leaf to kind of move on its own. In addition, there are other responses that aren't really locomotion based. It's more uh, biochemical, and a lot of the photosynthetic machinery tends to occupy this space. Especially certain things like carbon assimilation usually have very dramatic shifts in relation to time of day. Uh, where I really like to think about things, though, is that one thing that tends to happen quite a bit, since plants are sedentary and their growth is continuous, is that they're always sort of taking a bellwether of the environment around them, and they're modulating their growth in response to the environment that they're seeing. And odds are everybody in this room has at least thought about this once. And I'm going to provide you a example with this little ditchweed that could, Soteria viridis. If we were to look a little bit earlier on in its life history, it would have this lovely little shoot meristem. However, at some point, evocation is going to take place, and that shoot meristem is going to become an inflorescence. Uh, and once that inflorescence forms, that's usually a pretty good marker for about when flowering time is going to happen. And so, whenever you're measuring flowering time, you're actually looking at a trait that is developmentally derived. Uh, there's some other really interesting things if you like to think about modeling that plants also do because of this sort of regular repeated growth. One is that there identity is positionally defined by the cells around them. And one really great example of this is phyllotaxy, where if you're looking at the ribs of a cactus, or if you're looking down a maize ear meristem, you can sort of see these spirals of this geodesic architecture because you're just repeating the same module over and over again. Another thing that's really nice to think about once you kind of realize that these systems can be geometric is that a lot of people that do genetic mapping aren't just measuring one trait, they're measuring a couple. And I like to go hunting for these papers wherever I could find them. This Kumar one is just a good example, where you'll have these complex traits that are either related to something agronomic like harvest index, sometimes they're related to fitness or other sort of performance traits. But if you have other traits that are related to morphology and development, you can start to envision how there's some sort of sequence of events that gets you that same QTL that's silhouetting across these different genetic maps. And if you like to think about function value trait modeling, you can start to think about how these things could actually become functions of one another. And while all of this is taking place, it's usually probably the most boring time of the season for a lot of people where, you're kind of looking out and your plants don't really look like they've done too much, but they're actively making a lot of decisions in very rapid order. So just to kind of pivot now to thinking about stomata and making a case for why they're a great model for a complex trait. The first is that they're really easy to measure. So this is just a cleared leaf, and you can already see just the contrast of the cell walls alone. We can easily spot where the stomata are. The other thing that's really handy is that they do have a pretty key relationship to some important traits, especially leaf gas exchange and water use efficiency. And there's a great meta-analysis that another member of my lab, Daniel, had done looking at this sort of relationship between reduction in stomatal density and leaf gas exchange. Why I would actually argue that stomata are the most important, though, is that they are actually driven very precisely by some very key developmental steps. But before we go completely off onto that, I do want to just point out that grass stomata are a little funny compared to other stomata that we might see in dicot systems. So here we're going to look at an Arabidopsis, and they both agree on having the one key feature that a stomata needs to have. Stomata have this little pore, and that helps provide some permeability in the epidermis that allows for gas exchange that's needed for photosynthesis to happen. This pore is created because stomata generally have these two guard cells that are symmetric, and they can open and close. However, grasses tend to have these additional subsidiary cells that help them open and close faster. And there's some thought that that's actually kind of one of these novel features that lets grasses respond quite quickly. So if we were to now think about how we get to that sort of feature, one of the first things that happens when we're looking at stomatogenesis in grasses is that we have this establishment of these stomatal files that are regularly spaced and grow in a linear sequence down the length of the leaf. This is actually due to local inhibition, where these files seem to know where one another are, and they try to establish at least some set distance apart. Once all of that takes place, though, these key cell divisions kind of go on a rail where we'll have these fairly boring file cells that will undergo one asymmetric division, producing that guard mother cell that's going to be cytoplasmically dense. That then tends to pull its neighboring nuclei over in order to generate subsidiary cells. That guard mother cell then will itself divide, and then we just have cell wall modifications to kind of get to that final stomatal shape that we tend to think of in grasses. And so we can already sort of see that although this has implications for performance when it's mature, there's a lot of developmental biology to kind of get us to that point. And this is kind of where we enter thinking about some old data that was generated in our lab a couple of years ago in order to generate this new spatial patterning phenotype. So this work is actually derived from a B73 by MS71 rill population. It was grown over 2 field seasons during 2016 and 17. We largely focused on the 2016 data. And uh, this optical tomographer was used to generate these epidermal scans very rapidly. And then MassCAR CNN pipeline was used in order to annotate these epidermal cells so that largely count and size data could be generated for thousands and thousands of micrographs. So G et al. Largely focused on the sort of scored traits that were easy to pull out of these masks, largely cell size, cell shape, cell number, and they were related back to leaf physiology measures. And so although we know that stomatal patterning has some really clear anchor points in developmental biology, that really wasn't touched. And so joining the lab as a developmental geneticist, I just saw a lot of room to play with some old data. And the way that I approached this was I tried to create this stomatal neighborhood, is what I ended up calling it, where if we were to just look across this micrograph, we can see that there's stomata everywhere, but we're just going to grab 3 to start, and we're going to call these our origins. Once we kind of know where our origin is, we're going to reach out and find our first nearest neighbor, and then our next one, and then our next one. And we repeat this a number of times until the signal tends to dampen once we get to the higher orders. But once we kind of know where all these vectors are, we can layer them all on top of each other, and we can start to see that they are occupying certain regions of space preferentially. If we do this across all of a genotype like B73, we can already see that there's a lovely geometric pattern that seems to be emerging in this scatter plot. But it's not quite there yet for us to make a homologous comparison with just hundreds and hundreds of points in space. So we need some way to transform this into something that we can actually compare. The way that we did that is we ended up just creating a spatial distribution, viewing each one of these individual ranked neighbors as a probability. And so if we create this kernel density that describes the spatial probability, we already can sort of see that we get this nice geodesic structure that really does seem to be capturing the file system with those 2 hotspots to the left and right. And so here we're looking at it in B73, but there is, of course, the question, well, does this vary across our real population? And if it didn't, I probably wouldn't be talking here today. But we can already sort of see that B73 and MS71, they look quite a bit like each other. There's some slight differences in the intensity of their signal. But when we start looking at the rills, they are very striking at some of the phenotypic extremes. And not just striking in terms of like the intensity of the signal. If we just overlay approximately the area that B73 is occupying, and we relay that over the other genotypes, we can actually see that it's not just the intensity of the signal, but how much space they're occupying actually does seem to vary in this kernel density phenotype that we've created. So we already have a hunch that we're onto something. And the next question is, well, does this relate to any of the traits that Kevin that G measured in their study? So this is already kind of a bit complicated to handle since we're now dealing with a 2-dimensional distribution. So one way to handle this is we just flatten it across its 2 major axes. And now it's a little bit easier to handle. And if we then generate these flattened distributions for the entire population and color code their ranks for a trait like stomatal length or stomatal width, we can sort of see that maybe there's a little bit of stacking, but really those rankings don't seem to track very closely with how these distributions are overlaying. Now, if we do the same thing with stomatal density, however, these distributions almost perfectly seem to capture a gradient. And so it seems like this spatial probability is doing a very good job of capturing stomatal density. Generally, we use these sort of averages of the first five nearest neighbor ranks. There is some extra information, though, if we actually look at the individual layers. Uh, so the first two nearest neighbor ranks that you're seeing on the top there, those are overwhelmingly explaining our longitudinal information. So you could sort of view this as the file system of those cells kind of being printed out down the length of the leaf. However, as we go into the higher neighbor ranks, you generally start jumping over and capturing this lateral variation of how far neighboring files are from each other. Uh, One thing that people might have noticed if they were a little bit eagle-eyed on that first neighbor rank, though, is what are those little hot spots there? So that these little minor regions we're actually segregating in this population. And here we're looking at two genotypes that are sort of the extremes of what we're seeing. And as we look closely at the micrographs, one thing that starts to become clear where those four little hot spots are emerging is that you tend to get the files are so far apart that it seems like the file system gets confused. And so you end up with these sort of zigzagging adjacent files where they're kind of competing with each other because they don't really know where they should go anymore. Uh, so we already kind of knew that this was going on. But there were also a lot of other traits that we ended up wanting to pull out of here as well. Some already existed. We already had the stomatal length and width from G's paper, which corresponds to the little white rectangle in the middle. We also ended up capturing what we called probability or signal traits at three key positions in this distribution. Uh, the first was the in-file signal, which are those very bright spots immediately to the right and left. We also had this side file signal, those two fainter peaks above and below our origin. And one thing that actually becomes really stark as you look across a lot of these is there's always this trench that seems to cut off the side file from the in file signal. And that gap was actually pretty important as well. Uh, Once we kind of knew approximately where those foci were for the in file or the side file, cell spacing also seemed like it could be important. And so we captured our intervening cell number as a sort of cell normalized distance to the in file signal or the side file signal, respectively. In addition, we did also capture some variances because you could think of each one of these hotspots as being its own distribution. Uh, this did sort of beget a question, though, where we're creating a whole bunch of traits. Odds are a lot of these are co-correlated. How many are actually kind of necessary to describe stomatal density? So we tried to take a prediction modeling approach to see at what point we kind of plateaued on describing new information that underlies stomatal density. You can sort of see that during the first phase of this iterative modeling, when we just use cell size, we get a weak R squared of about 0.4. However, as we include our signal and our spacing variation, we very quickly creep up to explaining about 74% of that information underlying stomatal density. However, we pretty much plateau from this point onward, where we get a little bit more information by including those variant traits for the individual hotspots. But it's not really offering that much information relative to the model complexity. So we ended up cutting things off at that sixth model, where we're including both the signal and the spacing traits. And so one of the first things that I wanted to think about was if we could use structural modeling to try to capture uh, the patterns of interaction between these traits, both between themselves and between stomatal density, and then trying to ask, can we sort of see signatures of developmental sequence in those interactions? Uh, and so the way that I approached this was I used a directed acyclic graph using a piecewise structural equation model and basically optimized on the delta AIC of different model designs. What we ended up creating was this, which is a little bit unwieldy to look at, but we're going to kind of walk through the key parts of it shortly. But you can already sort of see that at the very top of this model, what it really kind of comes down to is that cell size and cell spacing seem to be driving a whole lot of the process. And ultimately, we're explaining where the variation from stomatal density is coming from. One of the first things that stands out as we're looking at the strength of those edge coefficients is that file spacing number alongside stomatal complex width and the trench signal seem like they're really important early sources of variation for where a lot of the signal is coming in for this model. Once we kind of enter where that trench signal is, we enter this region that I've taken to calling the triad, where we have these two paths that seem to be at odds with each other. The trench signal seems to have an extremely negative relationship with the in-file signal, and yet it does seem to increase the in-file signal through indirectly through that side file. And we're still like mulling over the implications of this, but given the fact that you have these two paths that are almost sort of mimicking a negative feedback loop, one thought is that this could allow these nonlinear patterns to emerge within this trait space once you kind of recognize that it's there. Finally, once we kind of get down to the in-file signal, it becomes really clear that the file system is a really important source of variation for explaining stomatal density. And so at this point, we have a pretty good idea how our trait space works. And this is a lecture series that is about genetics. So let's think about how some of these traits are actually layering onto the genetic map for this population. One thing that stood out immediately is that when we just did a QTL map of stomatal density, we recovered all of the same QTL that Kevin did in his original study. And we can already kind of see that stomatal density is highly polygenic within this mapping population. However, we're going to focus on two QTL, one of which looks kind of exciting over on chromosome 9, and one that initially might look a little bit boring over on chromosome 1. And we're just going to compare what information these two things are telling us. So as we're looking across our QTL maps for our signaling triad, if we just start by looking at the WAD map for the in-file signal versus stomatal density, it's very clear that there is a corresponding QTL peak where they both seem to agree that they really have a strong association with the same region around 100 centimorgans. However, as we start to look further up that triad, what we see is that there's actually an incredibly strong peak for the trench signal on chromosome 1, which is also shared by the side file signal, and it corresponds to that one peak that we might have initially thought was kind of boring on chromosome 1. And so it looks like we're already sort of seeing this segregation of our lateral and our longitudinal variation across discrete QTL within this population. As we sort of work our way back up a little bit further on the structural model, here we're now looking at our cell size and our cell spacing variation. If we begin with our lateral variation and what's feeding into the in-file signal, what we end up seeing is that that region on chromosome 9 that we're seeing does not have a corresponding peak for cell size with stomatal complex length, but it does for intervening cell number. So we're still not exactly sure what that region is doing, but it seems like it's affecting the spacing. In addition, as we now move a little bit further up and think about where that trench signal could be coming from, it is actually kind of lighting up across the board where we're seeing it both for stomatal complex width and file spacing number. And oddly enough, it's also lighting up for the intervening cell number. And this is one of these sort of takeaways that we actually got during this study, because it does seem like there is a hierarchy to how certain processes can emerge in development, where you can have these lateral QTL that affect the file spacing that can cascade into the longitudinal effects, but you can't have the inverse. And so this might have implications on how gene stacking might work. And at the risk of providing a dangerous analogy for this sort of thing, you can sort of think about developmental phenotypes as having a kind of architectural constraint to them. If they're all kind of doing loosely versions of the same thing. So if we have two key features here, a arch and a dome, there's maybe some feature that they agree on, but you need something to support the dome for you to have a dome. And so although you're looking at two different things, one of these is more dependent on the other. And how this might look if we sort of superimpose what we're looking at with our stomatal densities, we might actually start to think that, well, this file system, it isn't a strong direct effect on stomatal density, but it is having a knock-on effect with helping to establish the file system. So we already kind of felt pretty good about this story, but there were also some weird things that we were not expecting at all when we ended up looking at this trait space. The one of the main ones was, as we were doing ordination PCA of all of our different phenotypes, one thing that started to stand out is within this multi-dimensional ordination space, specific leaf area perfectly tracked with the trench signal, which I have done a lot of PCAs and I have never kind of seen something that is perfectly tracking when these traits have no relationship to each other, conceivably. And for those that don't think about SLA very often, this is basically just leaf area normalized by the dry mass of that same area. And so this is kind of a generic way of measuring the average leaf density. And so if we start to circle back and think about how the broader leaf was put together, here we're going to look at B73 and MS71. We can sort of see that we have these trenches that we already know to expect. And if we look about where those trenches are, one thing that starts to stand out is in a couple of these cases, these trenches seem to be occupied by these fiber cells that are running down the length of the leaf. Now, this is probably important in especially a C4 system like maize, because they are sort of overbuilt to optimize shuttling distances because they're C4. And so actually, if we're just comparing B73 to Brachypodium, one thing that's very clear is we have a lot more veins in approximately the same area. But also, if we look at those two stomata on the bottom and we sort of superimpose where that trench is, already we're seeing some bit of an internal anatomy that seems to correspond to that same region. And actually, this becomes even more stark if we flip the leaf over 90 degrees, because we can start to see that we have these files running down the length of the leaf, and about where each one of those trenches would be in our SPP phenotype space is where we would expect to see a vein. And so we're still not sure if this is going to track perfectly across other populations, but it was kind of a bizarre thing to realize that you have this sort of allometric trait that actually seems to relate to this probabilistic developmental metric. And so just to kind of summarize what I've gone through just now, we ended up recycling and sort of repurposing some old data to think about not just stomata as some sort of scored trait, but thinking about how we can generate some sort of spatial measure that can capture morphology. And in capturing that morphology, we could sort of think back on how the developmental sequence could actually have produced this phenotype that we ended up creating. We used that phenotype space to generate a number of traits, and we then explored them through structural modeling as well as through QTL mapping. And finally, we also at least got hints that there might be other parts of uh, leaf development that we're sort of seeing are like hidden under uh, these other traits that we really had not anticipated until we actually started on this study. And I feel like it would be wrong if I wasn't the one asking a question at one of these things. So I just figured I would like throw a general question out to the audience. Uh, Odds are we're all working on a whole host of different traits. And I can't help but wonder if more of us were thinking about what role development was playing in our phenotypes as we were looking at them. What new insights could we actually glean from this? And what could we sort of learn about how these plants are putting themselves together? And so, with that, I just want to go ahead and thank a number of people, especially members of my lab. Andrew, Jia Yang, Joe Crawford, James, and Daniel have all been great. I got some really great feedback from Toby and Michelle at various points during this study. This project was funded by the DOE and from a generous gift from Tito's. And the preprint is currently available, and I am actually going through the proofs for the Plant Phenome Journal. So the article proper should be forthcoming very, very soon. And so with that, I will take any questions.
All right, thank you, John. We have a question already in the chat, and then Jeff can unmute and fire away.
Yeah, just simple naive question. How have you looked to see if there's overlap with known GWAS or QTL hits for leaf morphology traits with the QTL that you found?
Uh, so I feel like that's actually a good point where I think we got so wrapped up with just trying to like work through the basics of like this spatial phenotype that I had not actually tried to go through broader like diversity screens, especially I think one issue sadly with my funding sources is that I think the DOE would really rather I worked on sorghum than corn. Um, but yeah, I think it's not something that I have looked at yet, but it is something that I think is definitely doable. Um, yeah, sorry, I wish I had that answer offhand.
Ruben has a comment. I don't know.
Yeah.
I was going to say something, Ruben. You're muted.
No, I just wanted to share that picture. Um, John, this is pretty great, and it's very fun and interesting how you have went back and how you go back and reanalyze these pictures. I was wondering, do you have a feeling how these traits then would change? For example, when plants are being water stressed and things like that, things that are known to affect stomatal density and other stomatal features.
Yeah, well, and we didn't actually. So everything that you're looking at is just a single field season. I think we did actually, although none of this actually made it into like the paper proper. Uh, we do have all the same analyses for the 2017 data, and one thing that actually kind of stood out right away is that you start to see that some of these QTL that looked like they were explaining variation in the 2016 season, 2017, the spring rains didn't come in Illinois. And so eventually they irrigated, but clearly the maize had already expressed that it wasn't happy that it didn't get rain when it wanted rain. And when you start to look at those QTL maps, one thing that actually happens is that file spacing number trait that we saw on chromosome 1, it actually seems to disappear because everything in the population starts to push its files as far away from each other as possible. And in my mind, it really just as I was sort of like thinking about some of these processes, I feel like a lot of development is probably where some of these G by E patterns that we're sort of seeing are coming from, especially the morphological ones. Uh, I don't know if that completely answers your question.
Yeah.
But
No,
yeah,
thank you.
yeah.
Yeah, I guess following up on that, John, yeah, and maybe you explained it and I missed it, but have you looked at if you just take one plant and you look at these stomatal phenotypes on different leaves or at different leaf ages or at different leaf sectors, like does that matter or is it very consistent across the leaves?
I started trying to do that. At least the patterns that I saw when I was doing this with a couple B73s that Joe Crawford had loaned me, it seems like the juvenile leaves are a little bit inconsistent. They don't quite know how to put themselves together. However, as you start to produce mature canopy leaves, you start to get homogenization of this phenotype. So they're kind of building a messier version of it earlier on. And then once they kind of get up to running speed, then they start to produce these really nice geodesic patterns with regularity. Um, as for how that would vary across different genotypes or different species, I feel like that's kind of that's the open question where I'd imagine that it must be varying and that there's probably some sort of trade-off for uh, building something really well that maybe takes a lot more effort on the plant's part, or just building something that's sloppy and good to go. Uh, but I feel like that's actually what I don't know the answer yet, but I feel like that's kind of what makes these sort of spatial phenotypes really exciting because there's a lot of ground left to cover.
Yeah. And so like in terms of like the bigger picture, do you know or do you hypothesize like what kinds of stomatal phenotype or structures are I don't want to say optimal, but better adapted or better functioning in different um, ecologies? Do we know anything about that?
Uh,
Something about that?
I uh, I think generally, at least from some of the work that's been done using transgenics in our team, usually having stomata that are sort of hypersensitive and having fewer of them is the way that you kind of maximize water use efficiency by trying to reduce leaf gas exchange. There is, however, a trade-off there where, in theory, if you're making a leaf that is barely permeable, it's also now barely permeable. And so if you're now sort of limited by how much CO2 you're taking up, that might have a negative impact. C4s are one of these few systems where they kind of cheat already. If they're already sort of enriching the amount of carbon dioxide that's in their leaves, in theory, they can get away with having fewer stomata just fine. But I think a lot of C3s, there is probably more of a constraint where if you overbuild uh, the system where you have leaves that are barely permeable, that actually just kind of decelerates everything. Um, but yeah, we do know that this relative arrangement does vary even between maize and sorghum. They're not exactly putting these files in the same spot. Uh, but I think that's where, as sort of a developmental geneticist, thinking about like what genes are turning off and on to kind of regulate that relative patterning, uh, there's a lot of fun ground left to explore in that space.
Yeah. Okay. Couple more questions. So Jeff has another
Yeah.
picture. I guess folks can look at that about teosinte and trees and Jagdeep. I don't know. You're able to ask. Yep.
Yeah,
Go ahead.
I can ask it. Yeah, this is very cool. So I study roots, and cortex has these beautiful, cell layers, and I wonder the same things as you do here. I was wondering, like, if stomatal density is your optimization function,
Yeah.
and you're trying to optimize based on water availability, let's say, do you think plant is making choices to go with this feature or that feature in terms of being a proactive measure versus a reactive measure to the water availability, or even the VPD in the atmosphere, right?
Yeah, on at least as these things are first forming, so that those developing stomata that I was pulling out of Soteria, usually the sort of core of the culm where the meristem and the developing leaves are, that's almost an aqueous environment. Like as you cut into them, they're actually sort of, it's saturated. And so they are kind of getting probably some sort of a signal from the mature tissue that's above them. So I don't know if they're responding to the environment so much as they're kind of hearing the leaves above them screaming and then they're responding in kind. Uh, yeah, I do think that there probably is some trade-off where, uh, oh, actually, here, let me see if I can share one of my slides. There we go. So this was actually from the 2017 data that we never actually ended up publishing. But you can sort of see that that region on chromosome 9 is still kind of doing almost what it wanted to do. But the chromosome 1 QTL is all but gone. And what it does kind of seem like happens is the year-over-year variation is mostly working off of that file spacing number, where you kind of have like a couple genotypes that are sort of panicking even during a good year. And so they just always put their file systems as far apart as possible. And then that creates a very disordered system. Eventually, once everybody gets hit with enough stress, they all start doing that. However, in my mind, that's actually what kind of makes these few intervening cell QTL all the more important, because that's clearly the limited source of genetic variation, at least in this phenotype space, because in theory, you probably only can have very mild defects to the file system, or you don't have stomata. And once you don't have stomata, you probably have other problems. Yeah.
Okay, thanks. I think that's all the questions. Thank you, John. Cool stuff. And we'll see everybody next week. I think we have Mauricio Bayon from Arizona State will be presenting next week. Thanks, everybody.